Welcome, panelists. Hello, hello, welcome. I am so excited for this conversation today. As you're joining in this webinar, there is a chat button. We would love for you to welcome yourself into this space. Go ahead and share your name, introduce yourself, where are you in the world, and what are you excited to learn about today? Come on in. And as you're getting settled in, I am based in Portland, Oregon, Pacific Northwest. I am really, really excited to get my plant physiology brain, my secondary metabolism brain excited today. Water use efficiency, talking about all kinds of things, all things leaves, all things flowers, all things fruit. I'm really excited. And again, my name is Dr. Adrienne Gottschalks. Welcome, everyone. Come on in, introduce yourselves. Where are you based? And what are you excited about for today? What about you, Brian? What are you excited about today? And where are you now in the world? Since it's new.
I'm a traveling man. I just moved from California up to Oregon, and my wife and I are living out of a fifth wheel while we're searching for a home up here. And yeah, it's different. It's definitely a different climatic environment from hot and dry to I think rainy and wet so far. But my impression so far have always been in the winter time frame. So
yeah, you did manage to get here just as those atmospheric rivers started to unload. We got 6 inches of rain in Corvallis last week. So whoa, my poor wheelbarrow was overflowing. That hasn't happened for a long time.
That's impressive. That's great.
Yeah, it's been raining almost every day, but that's good. It's something different, that's for sure.
That's for sure.
Ooh, I want to give a shout out to a couple of my students. Marcus, Philippe, thanks for joining. Great to see you all here. As a mentor for the CTP program at the Soil Food Web School, I get to work with folks who are out there helping plants grow healthy also. So it's really fun to see that they're joining this as well. And I see some names from our FC forum, so it looks like we have some foundation course students joining. Oh, Todd Harrington, welcome. Great to see you.
Hey.
It's great to see the diversity of places. We have people from Brazil and from Turkey, from Australia, from just all over the world joining in today. So that's, I think it is a foreshadowing of the need to find out about how people can grow plants without causing toxic chemical problems. Portugal. All right.
Yeah, I think
Nigeria.
we hit all continents again.
Yep.
Still waiting for the Antarctic contingent to show up. But,
hey, I had a friend who just went down to the Antarctic. So what are they doing? Not saying hi.
Oh man.
Exactly. They should join and then we complete the circle, right? We've got
some enthusiasm about learning about how to regenerate dry areas. So that's something we can talk about, especially around plant health, water. Big deal. Great. Excellent. Yeah, so if you're just joining, go ahead and enter your name in the chat and welcome yourself. Thank you for being in this space with us. Where are you in the world and what are you excited to learn about today?
Another Aussie.
Awesome.
France.
And John, what are you most excited to learn about today?
Well, I'm looking forward to a question that I'm going to ask both Elaine and Brian, and I'll give you a bit of a heads up. And the question is, what perspectives or opinions or observations do you have that are distinctly different from those of your colleagues or peers?
That's a great question. Okay, don't answer yet. That's the teaser. Right. That's the trailer.
I thought about that question. I thought, now if someone were going to ask me that question, I would like to have a little bit of a heads up. So
that's fair. That is very reasonable.
That's a thinking question. That's definitely a deep question. Yeah.
Wonderful. Okay, so I'm going to go ahead and get this slideshow rolling so we can get to some of those fun questions. So this is a brief overview of what we're going to do today. First, I'm going to tell you what we're going to do today. So let's do that. The quick housekeeping around our Zoom. This is a Zoom webinar, so we're grateful that you get to be here in this call with us or watching on a YouTube Live or wherever you are. But if you're in the Zoom with us, please enter all of the conversation that you want to in the live chat. This is a space for you to come together with one another The Zoom Q&A is what we're going to be paying attention to closely and filling in questions, so that if you have specific questions that you want to ask our panelists and have talked about live, we can answer those. Or if we don't get to them, we'll make sure to try to follow up with you. And above all, this is a space for us to all grow the healthiest plants possible and create a beautiful community that supports as many people as possible. So I will ask that we have respectful language that can take care of this community that's out there doing good work. So if there's anything that I need to address, I will interrupt our conversation and talk about it. Okay, so first, the Q&A, the icon down here looks like 2 different chat buttons. That means you want to talk to us live, and the chat is where you get to talk to one another. We don't pay as much attention to that for specific questions for this call, just so you know. Here we are, welcome. Okay, so welcome our panelists. We have Dr. Elaine Ingham, my personal mentor and the soil microbiologist who did the foundational work that has led to us creating the Soil Food Web School. She is a, Robin Hood, taking this out to the people and getting it out of that academic ivory tower, and I'm very grateful for that. We also have John Kempf today, who's a leading crop health consultant, self-taught, brilliant, interesting conversationalist, and I cannot wait to engage further, and he'll tell us more about that as well. We have Brian Vague, my colleague, fellow CTP mentor, also a soil food web consultant who is helping farmers grow healthier, more drought resilient almonds and oranges that are pest resilient and other factors that you'll hear about today. And my name is Dr. Adrienne Gottschalks. I am a chemical ecologist at heart, which basically means the compounds that a plant makes mediates their interactions and collaborations with other organisms, whether it's an invertebrate, a wasp, an ant, a microbe, any symbiotic relationship to create the big network of relationships that plants maintain at all times. And with that, let's get started. So first, I'm going to ask each of our panelists to briefly introduce themselves and their conceptual theory that they spend most of their time in their day thinking about. So I'm going to pass that to John Kempf. Welcome.
Thank you. I'm delighted to be here. Thanks for inviting me. I've been looking forward to this conversation. I'm not sure how to describe the scope of the work that I've been passionate about for the last decade succinctly. So I grew up on a farm where I observed agriculture being incredibly destructive. And we all know and understand the scope of some of the damage that we're wreaking on the ecology around the planet with our agricultural practices and industrial practices and so forth. And I soon came to learn that when we have really healthy plants and a really healthy agriculture ecology, agriculture does not have to be the source of so many of our pollution problems, but it actually can be the solution. And I became really inspired by the idea that we could grow really healthy crops that were so healthy that we did not need to use pesticide applications and reduce or completely eliminate the need for pesticides, that we could have a tremendous positive impact on public health by producing food as medicine, and that we could regenerate soil health and regenerate ecology and eliminate nutrient pollution and runoff, et cetera. So my personal passion is having these regenerative models of agriculture become the mainstream around the globe in the next 2 decades. And that's what I'm here to accomplish.
Yeah. Do you have anywhere in your materials from pollution to solution?
No, I
Because
don't.
that's just what rang true in my brain from what you said.
I like
Lovely.
that.
Wonderful. Thank you for being here. Also, John has founded Advancing Eco Agriculture, which is both a consulting company and also a community of folks who talk about these things. And there's a forum that you can belong to that I hear wonderful things about. And this is John's plant health pyramid, which, do you want to speak about this for a moment, or should we come back to this?
The plant health pyramid evolved around the observation as we worked with many different farmers and different crops and different types of environments. We observed this gradual evolution of plant health where plants, as they developed their nutritional integrity and microbiome integrity, they became more resistant and increasingly resistant to different groups of diseases and insects. And so we developed the pyramid to describe some of what we were observing. And there are hundreds of peer-reviewed papers that support the science behind this. And I've given several presentations on this because it's too much to get into in just a few minutes. But the foundational idea is that at the beginning, at the bottom of the pyramid, once plants have the nutritional integrity to produce high levels of photosynthase, higher than what is considered to be average or normal or common, then we get two things happening. One is we get a larger quantity of sugars, and we also get a, if you will, a higher quality of photosynthesis and more complete carbohydrates being developed. And at that stage, you get resistance to all of the soil-borne fungal pathogens. And the second stage is when you have complete protein synthesis. And these first two stages are purely a function of properly balanced nutritional integrity, mineral integrity. However, the latter two stages are a function of microbiome integrity. When we get to level 3, plants have this surplus of energy that they store in the form of various lipids and essential fatty acids. And these are then the foundation for a broad array of different plant secondary metabolites that plants produce as a part of their immune function, as a part of their immune and various resistance pathways. So what we have observed, and this is this statement at the bottom of this infographic, healthy plants can become completely resistant to all diseases and all insect pests. And that's a very bold statement. It's a very big mouthful. And yet it's one that I'm completely confident making because we've actually done it. It's not a theory. It's not a hypothesis anymore. This is something we've actually executed in practice. And at this point, we've been invited to work on close to 50 different quote unquote incurable problems where there is no known chemical solution for a specific pathogen, such as Pierce's disease on grapes, for example, or citrus greening. Or crown gall. There's a whole range of these different organisms. And after working on 50 of these different diseases over the last 15 years, we have not yet encountered one that we were not successful in reversing completely with nutrition and microbiome management. So after that kind of a track record, I don't think it's displaying any hubris to say that we can control these challenges with nutrition management and with microbiome management.
Can we get a what?
Yeah. Exactly.
Yeah, Elaine, Brian, raise the roof if you've seen a similar thing. That's brilliant.
Yeah.
Thank you. I love it. Speaking of Dr. Elaine Ingham, would you care to introduce a little bit about yourself? And then I have an image of something that might look familiar to you.
Might be, huh? So I'm Dr. Elaine Ingham. I've been doing work in soil ecology, the organisms that make up the soil food web for 45 years. When does it roll around to 46 years? I don't know, but coming up on it. So I really try to understand the interactions of all of the organisms working in the root system above the ground on the foliar part of the plant, and the interactions with the plant. So I think you can see where the work that John is doing and that we do is pretty closely applied. You know, it's almost like we should be talking and working together on things, because what he's talking about, what I'm talking about, affect each other. And hopefully we can work in tandem and not at cross purposes at all. So I've been working with people around the world since 1985 to put this biology back into their soil and then see the benefits that, well, John was just talking about them, some of the benefits of getting this biology back into the soil. What I like to say to growers is we should have all of these organisms in the soil food web back in the soil because they've been destroyed. If you're putting out inorganic fertilizers, if you're putting out pesticides, herbicides, you're doing any toxic chemical applications, you're destroying this biology that's present in your soil. And this biology is something that nature meant for them to be there to do these processes for your plant. Well, if you kill these organisms, then you have to start to do their work because you've taken them out of the picture. And us human beings, we are really abysmal at understanding what's the nutrient that your plant needs right now. What's the nutrient that it's going to need? What's a different nutrient or the different nutrient that it's going to need once this current nutrient limitation has been taken care of? And how does that work? How do all of these moving parts affect your plant? So we talk about 7 overarching principles that if we can get the biology back into the soil, back onto the surfaces of our plants, then we as farmers, growers, don't have to worry about that aspect of raising the plant. And so really, if you think about this, the only time you're going to have to really be outside doing things is early in the springtime to make sure that the biology is in place, that we've got the organisms, the bacteria and the fungi and the protozoa and the nematodes, and then the higher level predators in the system like the macroarthropods, the earthworms, things like that. We've got to have all of that working together so that we can get all the nutrients your plant requires. There is no soil on this planet that lacks the nutrients to grow plants. They're all there. And one of the things we've been working on recently is replenishment of those nutrients through the growing season or through the dormant season. How do those nutrients get returned to your soil so your plant can continue to draw on these sources of nutrients? You can only convert those nutrients that are present in the soil from the plant not available form into the plant available form. The only way you can do that is by having this soil food web operating properly with the local indigenous sets of microorganisms present, so they'll survive from year in year. They'll be doing their thing from year to year. All you have to do is check up and make certain that these organisms are present in your soil. They're doing what they're supposed to be doing. You've got enough of them. What are the minimum levels? So understanding this whole food web is what the Soil Food Web School is all about, where we offer foundational courses for you to understand the science, for you to learn the critical parts about getting the proper biology back into your soil. When do you apply? How much do you apply? How do you know what your plant requires? So we go through all of those things. And then of course, if you want to start a career basically doing these kinds of things, the school takes you through being able to do samples of soil or tea or compost for other people. And then if you want to become a full consultant, we have a consultant training program where we're going to get you to think things through and figure out things for yourself instead of always having to have a pat answer handed to you. So we're continuing to work on this project, and we're starting to move out into more global representation, because you can't grow compost in one place that will supply the world. The transportation costs of getting that compost out to somebody in India or Brazil or wherever. You need to have local indigenous organisms. The organisms that nature has been working with for the last, well, let's see, bacteria were first found on this planet 4 billion years ago. So they've had plenty of time to get regionalized. And so we want the regional sets of bacteria. Well, how about fungi? They've been around for, about 3.5 billion years. And anything that nature is going to keep around, make certain that these organisms have survived for billions of years have to be doing something critically important. And so us as growers, us as people that want to make certain our children and our grandchildren will be able to live healthy lives, we've got to get this biology back into the soil, and we've got to get people understanding why it's important.
And that's a key piece to how we can have a nutritious plant as, sorry, the mailman, my dogs. I'm sorry, y'all.
No worries.
But that age, that evolutionary age, and the number and diversity of metabolism that has evolved in each of these different organism groups is part of the secret to how the soil food web can help us have a nutritionally complete plant. And so that's a little key teaser for what's about to come. But first, I wanna let Brian introduce yourself. Carry on and tell us a
Sure.
little bit about what you spend your time thinking about.
So I definitely feel like the new kid in the block. Elaine and John definitely have a lot more years of experience and knowledge than I do, but hopefully I can match that in passion and for what I do. But my history is that I came from the high tech world. In college, I was information systems management. I worked for a large IT company for 25 years. But I didn't kind of feel like my passion was being fulfilled. And in the background, I grew up on a homestead. My wife and I, we bought a homestead. We were very much into growing food that's nutrient dense, that's healthy. As John mentioned about food as medicine is a very important and dear thing to me. And when I first started my journey of my homestead, I grew like my parents did, which was very conventional. We rototilled, we applied, fertilizer, herbicides when needed, pesticides, those kind of things. And after doing it on my own homestead, I just started looking at the labels of the products I was putting on my plants and going, skull and crossbones, this is poison. Why am I doing this? This is crazy. And then seeing year after year, less production, more need for inputs and those kind of things. It was just a cycle that I couldn't keep up with, nor did I want to. And I came about permaculture to really kind of first start this, and look at the design sites and the interaction of plants and removing those agrochemicals from the system. And I was seeing really good successes, but I still didn't understand the whys. And I read a book called Teaming with Microbes from Jeff Lowenfels. And Elaine wrote the foreword to that book. And I just read the foreword and I went, oh my gosh, okay, this is really explaining a lot of things for me, tying a lot of dots. And then I just went head straight into the whole world of soil biology and the benefits that you get from it, applied that to my homestead and started seeing great successes. I took the training from Elaine. And then basically to my wife, I said, I'm going to go through a little bit of a midlife crisis here. I want out of the whole high-tech IT industry, and I want to do this as a living. I want to help, other people, particularly farmers, convert from conventional farming to, biological farming and farming, in regenerative types of systems. And so that's how I came about this. And I started my business back in 2017 and haven't looked back. And it's been a life-changing thing for myself, but also it's just amazing to be able to work with people and farmers and help them go through that transition and watch them kind of get exposed to the potential that they actually have on their own farms. So that's my story.
Brilliant. Thank you, Brian. I absolutely understand this life career shift. I was a career academic, if you will, going up the chain from grad school into becoming a college professor because I thought that was the only way that I could help people understand and appreciate the natural beauty in the world. And I have massive respect for that track. And I kept getting pulled back to working with and communicating with anyone. And I'm so grateful that the Soil Food Web has given me a way to empower people to directly impact and grow healthier plants and help that soil become healthier and more alive with all of the complete groups of the Soil Food Web organism. So it is amazing to land in a place that feels like, yes, this fits. So I'm glad you felt that too, Brian. It's so cool.
Oh yes, very much so. Yeah, I can't look think of any other path but forward with this right now. I mean, this is fantastic. So,
all right, welcome everyone. So just to see who has now joined on, who we are, you've heard a little bit about who we are. We want to know a little bit more about who you are. And if you've been in one of these before, we've shifted because we recognize that people can be multidimensional. So now there's several options. So in the poll, which, do I need to launch that, or will it automatically get launched? If I need to launch it, someone in the text, oh, there it is. Look at that. So there's 4 options.
You got launched.
That's perfect. We have, are you a farmer, grower, or agricultural professional? Are you an environmental advocate? Are you both, or did neither of those terms feel right for you? Go ahead and click on your best option, and we will see if we can come up with a soil pun in the meantime. Does anyone have a good soil joke? Oh, Elaine, I think you're muted. Hold on.
Yeah.
How long are we going to give them to choose?
As long as they want, take the next 27 seconds.
Okay. Yep.
Should take more time than that.
One of those paradoxes of life, right?
Take as long as you want within the next 30 seconds.
Yeah,
Well, we're now
true.
down to 15. So
That's true.
better get figuring that out.
So my favorite joke as a kid, I know it's not soil related, but what's the furriest side of the cat? Anyone? The outside.
Oh, you bum.
I don't know why that one
It
stuck
does.
with my mind. So,
I'm gonna
Um,
remember that one.
tech team, can we reveal our results? Or are we still rolling? It looks like we have a mix of folks. Many people are both. Some are just here as environmental advocates. Some are strictly growers. Awesome. Thank you all for being here. No matter what perspective you're coming from, I think it is important to make that paradigm shift that Brian's talking about. That is, why are we putting poison on our plants and expecting them to be healthy? Or how can we transition? So speaking of health, one of the things that I've asked each of our panelists to do is to think about what a healthy plant really is, but now I'm going to challenge them even further to, in whatever terms feel right for you, whether you sing me a song or write a poem or even just describe the physiological mechanism, what is the evidence or what image comes to your mind when you think healthy plant? So describe for me a healthy plant. And I'm going to let this be a free-for-all, so you get to call on each other and talk to each other. So carry on.
So I think I would describe a healthy plant as a plant that is producing exudates to feed the microorganisms in the soil to make the enzymes that that plant requires of them. So the plant will be able to pull those nutrients, those inorganic, not available nutrients out of the sand, the silt, the clay, the rocks, the pebbles, and of course the organic matter in your soil. And then those nutrients, all those foods are taken up by the bacteria and fungi, which produce those enzymes. And so the bacteria and fungi around the root system of the plant is gaining those nutrients. We've got to have the predators come into the system, the protozoa, the nematodes, the microarthropods, the earthworms need to come on in and eat the bacteria and the fungi that have these elevated levels of the nutrients inside their bodies. And because the nutrient concentration in bacteria and fungi are so much higher than the predators that are eating them, the predators release those nutrients in a plant available form. And so your plant can take them up because, of course, where is this interaction happening? Right there next to the root system. So the plant is immediately going to be able to take up the nutrients that it requires. If all of the nutrients are not taken up by the plant, then the bacteria and fungi that were not eaten by their predators can take up those nutrients. They'll still be subject to the next set of exudates that come from the root system, but you can see where this over and over, how many times a day does that nutrient cycling turnover happen? And some of the work done at the Duke Phytotron has shown that it's seconds for all of these interactions to be occurring and the plant to get the nutrition that it requires without us having to put on any inorganic fertilizers. And indeed, if you put out an inorganic fertilizer, you're going to kill these organisms in the soil that do this work. So now you're going to be obliged to do their work for them if you are audacious enough to go out there and kill these organisms that are working in your benefit.
So, Elaine, it sounds like you're describing that a healthy plant is defined by the organism or the community of organisms surrounding it.
Yep.
True?
Yep.
Okay. Great. What do you think, John?
Yeah, I would say first we need to think about, it's so common to frame this conversation in a binary context to say that a plant is either healthy or unhealthy. And it's too simplistic, I think, for us to look at it from that perspective. We need to recognize that there's a spectrum with varying degrees of health. And in that regards, it's very similar to our own health. So I've heard some of the craziest comments about people's health that you can imagine. For example, someone was recently diagnosed with lymphoma, but aside from that issue, they are perfectly healthy. And it's just such an oxymoronic perspective that it puzzles me. And so I had this interesting conversation with Dr. Arden Anderson, where he said that, we should think about levels of health in, at the very least, 3 different categories. First, you have disease, where you have an unhealthy plant or an unhealthy person where disease is already present. And then on the opposite end of the spectrum, you have the epitome of health, which is the complete absence, the complete freedom of disease. But then there is this range in the middle, which is generally in a pre-disease state. So disease may not be present yet, it may not be manifesting, but there is an environment present that is conducive for disease to manifest itself. And so when I think about what is it that describes a healthy plant, it is from a very practical, experiential perspective, a plant that is free from disease. And I would expand that to include insect susceptibility and nematode susceptibility and so forth. So when we have a plant that is free from pest attack, that can only occur when you have a plant that has an optimized immune system and that has complete microbiome integrity that Elaine described, that also has complete nutritional integrity and has the capacity to be resistant to whatever might be choosing to infect it. So when I think about what defines a really healthy plant, it's a plant that has the capacity to be completely resistant to all of these potential infections.
Really interesting, John. One image that's coming to mind is a emotional challenge I had when I had a bunch of aphids on my kale plants, but I also noticed as I was like brushing them off that there was a parasitoid wasp wandering amongst that community and that there wasn't a whole lot on there. So as I was kicking them off, I realized that I was getting rid of the food for my predators in that system as well. So when you say a pest-free, disease-free plant, is that one that has manageable communities that are healthy so that those food sources for predators are present, but just not in outbreak status? Or do you mean there's no insects whatsoever on that plant?
Well, I would say my experience has been that the presence of the population would be so small as to be virtually unnoticeable. So perhaps not completely absent, but to be virtually unnoticeable. And we've actually observed as plants go through this transition, this evolution where they achieve higher levels of plant health, at first, there is an abundance of predator species going after aphids or going after spider mites or whatever the pest might be, for example. And then within a year or 2, or sometimes longer, depending on the crop and the cycle that we're talking about, those predator species, those beneficials, tend to fade away and largely disappear, or at least they're not, we don't notice them anymore. And we don't notice them anymore because their food source has disappeared. The plants have become healthy enough that they are no longer a host for the aphids and the spider mites. And so because the aphid and spider mite population is decreased, that means the population of the predators that feed on them also decreases proportionally.
Great. I think you want me to go, Adrian?
Yeah, Brian, I'd love to have you. Adrian is having a bit of an emotional moment with the dog.
With the dogs, exactly. No,
Nothing
no,
to do
got
with
it.
aphids on kale plants. Dogs in the background. Yeah, hey, Brian, I'd love to have your
Like kale.
perspective.
Sure. So, you guys hit a lot of the high notes for me. One of the things I was, when I thought about this question was about the immune response. The plant has a significant immune response to be able to either not attract insects, and then also, to fight off diseases. But there's another thing that I also think about as well, and it's maybe a hard thing to describe, but once you get experience of growing a particular plant, you can tell when a plant is vigorous or in vigor, that the expression of the plant has the right coloration, has the right rigidity, it has the right growth to the plant. And it's more of an intuitive feel that you actually get. You can see that plant is in vigor while that other plant is not. And when you mentioned the talk about humans being sick and so forth, you can see that once humans, because we all are humans, that when, wow, that person doesn't look all that well. They kind of look a little bit ashy or pale, or they've got a
Green
gait
around the
that doesn't seem quite
gills.
right. Exactly.
Right, the
Um, it,
sparkle in
it,
their
it-
eyes, right? That, like, you can see the
completely.
moonlight in their eyes when they're just vigorous and vivid and
E-
alive.
Exactly. And, it was interesting. I was at a client, and she had a Scots pine that was being infested by a turpentine beetle. And so we went out and assessed the area, and it was primarily due to heavily compacted soils because it was next to a construction site and so forth. And we decompacted the soils, we added a lot of compost extract, and we did all these kind of remediation aspects to it. And when we first went out there, the tree was not putting out any sap. The beetles were having free reign inside the cambium, and just the way that the tree presented itself was kind of hanging down low. The needles weren't upright. 2 weeks after we did our treatment, came back, the tree was pumping out sap like crazy, and the needles were up. You could just tell the plant had so much more vigor. My client said the same thing. She loved this tree and had been around the tree quite some time, and just said, I could see the difference. And it wasn't just one thing, it was a many different things that added up to say, okay, this plant's bigger. And so for me, that would be the only addition I would add to that, is that when you see a healthy plant, you get experience. If you look for some of the signals and kind of get your observation skills out, you can start to see what that vigor actually looks like.
And
Brian, that's an excellent comment. And I would want to build on that to say that when you have these higher levels of plant health expression, plants also begin expressing themselves different genetically, or epigenetically might be a more appropriate term, where I've so many occasions observed that plants begin behaving differently and setting fruit differently. The branching structure begins behaving differently. And there's no good way to describe a broad range of crops succinctly other than to say that they start behaving differently. And the two archetypes that we see most commonly are that nodes become much more tightly spaced, plants become much more reproductive. They produce much higher for the plants and the crops for which that is appropriate. They tend to become much more reproductive. And as a general rule of thumb, there are exceptions to this, but as a general rule of thumb, plants and the individual plant parts tend to become shorter and wider as a proportionate ratio. So leaves tend to become shorter and wider. The stature of plants tends to become shorter and wider. And this is true not just of our domesticated crops, but also of the plants that we refer to as weeds, where if they are in an environment that is really conducive to their expression, they tend to be shorter and stouter. So there's lots of different pieces to observe there. That's an excellent point that you raise.
And I think there's an aspect of going beyond just looking at whether something is diseased or is not diseased. If they're not dealing with a disease-causing organism attacking them at this time because they're resistant, they're immune, or capable of dealing with it, there's a level of vibrancy, of additional production that plants can put on that as long as they're disease free, they're going to go ahead and express that additional genetic material. And we see it often in fruiting plants, because instead of just one apple, now we've got 2 or 3, and you start, looking at the whole plant, and there's a massive amount more production of oranges or apples or pears or whatever. When you've gotten to that point where the plant's not fighting a disease, its vitality is that much more complete. And in the commercial world, in the chemical world, most of the time they would go through and they would thin those trees, removing the extra fruit that's being born, because they think the plant's just not capable of it. Well, if we get the biology back into the soil, if we get that plant back into a healthy condition of not being diseased, where it's got all the nutrients it needs, you can harvest so much more fruit from that plant, going beyond that reliance on it is diseased versus it's not diseased.
I think
Sure.
it's
And
important for us to go ahead, Brian.
I was gonna say, the plant's got, it's built that structure, it has the ability to support that heavier bearing fruit load and so forth. Whereas if you have a weak plant, I mean, you go out to orchards or other kind of growing situations, and if they don't thin or put too much bearing because they pump too much of a particular nutrient into the system, then you get breakage, you get other damaging aspects to the plants. Yeah. Sorry, John, go ahead.
I think it's important for us to also recognize that most of us don't have the experience or the context to even understand what truly exceptionally healthy plants actually look like anymore. We don't know. We've never experienced it. We've never observed it. And I didn't realize this for some time when I was first starting out on this journey, but we have now had such a number of experiences in different crops. And I've posted some of these on my blog and on Instagram and different places, but we've been able to grow radishes from seed to harvest in the typical 6-week period, where the radish size is the bulb itself, not the entire plant, but the bulb itself averages between 8 and 10 ounces. And they're sweet and crisp and clear all the way through. They're not woody, they're not hard, and they've not become hot. That's normally you grow these are the exact same radish seeds that normally grow to the size of a quarter in a 6-week time period. Now they're the size of a softball. And there are so many expressions and so many examples of that, where we've grown peas where the leaves are the size of your palm, and green beans you have half a bushel of green beans per plant, or soybeans where you have 12 beans per node. And so there's so many examples of this, that once we really achieve these upper levels of plant health, plants will begin behaving in ways that are dramatically different from what we think of as being common or normal. And we're going to have to adapt accordingly.
Yeah. Great.
And your description of these different plants is bringing up an image for me. I asked our mentor team this morning the same question, and the overwhelming themes that came out were whatever that plant is, that plantiness of it. So like walking through a tomato field, smelling that rich summer tomato smell. And there was a while that if I were still in the business of writing papers to get a job, one that I would have loved to write right now is, what is the defense ecology of a healthy plant? Like, what is the secondary metabolism allocation into different leaf ages, different developmental stages of a plant that's healthy? How much are they investing in recruiting those predators, in root tissue? I want to give a shout out to Janet in the chat, who said that a healthy plant has deep roots, and I could not agree more. But I believe the health of a healthy plant is almost an emergent property that makes that plant, as like the green bean, have the delicious beaniness that we know to be a green bean. And that's the secondary metabolism that's at that tip of that pyramid, right? So let's talk a little bit more about how we get there now that we have this, unless anything else is coming up for each of you.
There's one more point that I want to add, which is the domain, the practice of regenerative ag is entering into the fringes of mainstream science and achieving more scientific credibility, more scientific research is being done, which is all very much needed, necessary, powerful. However, there is one consistent flaw that occurs repeatedly in most papers, which is there is no consideration given to the degree of plant health of the plants that were used in the study. So when we're testing the effectiveness of a microbial inoculation, or if we're testing the degree of insect resistance or disease resistance based on a certain nutritional profile, we actually have, or if we're testing carbon sequestration and the movement of different, as you described, the partitioning of different exudates to soil profile, we know that the literature reports wildly varying degrees of what percentage of plant carbon actually gets sent out through the root system as root exudates, ranging from a low of 7 to a high of 80+% that is reported in the literature. And the question we should ask ourselves is, why the variability? And there's a number of reasons for that, obviously. One is microbial ecology, but another significant contributing factor is the degree of plant health. And so we need to keep this in mind that many times growers are reporting amazing results, being able to sequester carbon and build soil organic matter very rapidly. And scientists are saying, well, wait, it's not possible. The literature doesn't support the possibility of being able to sequester carbon that rapidly. And the farmers are saying, well, I'm doing it. I've been doing it for a decade, so I don't care what the paper says, it's working. It's how do you dispute the reality of what actually exists? And this is where the discrepancy is coming in, is that from one significant discrepancy from my perspective is that the research science does not take the degree of plant health into consideration during the research. And Elaine, I'd love to get your perspective and response on that.
I'd have to think about that one for a while. What is it that we could be measuring? And there's some of the newer measurements that people are taking, like assessing the materials in plant sap. It's much faster way of assessing what's going on inside that plant from a physiological point of view. Have you reached that stage where the plant has overcome any requirement to respond to diseases because it's immune, it has the ability to deal with those sorts of things and not be sidetracked? And so that's the point at which we want to be doing all of our testing. Or do you want to start all of your research work with not very healthy plants? So you can document that by addition of this or that microbial aspect, these plants get better. The ones in the control does not, because there's no way that the beneficial organisms can come back.
Well, as an Elaine understudy, from your definition of plant health being shown and exhibited by the community of microorganisms that surround that plant's roots, cover those leaves, it does all start with photosynthesis, because photosynthesis is, I think, the most beautiful biochemical reaction, but we're not here for philosophy. So I'm going to jump to, instead, it drives those exudates, as we talked about. And it is the beginning of what fuels the food web that is the source of all of those metabolic pathways that can extract and mine out the nutrients. And I completely agree with you, John. I think that if every paper that I read also included how many protozoa and nematodes were, beneficial there, supporting the plant in order to consume the bacteria and hyperconcentrate the nitrogen, phosphorus, potassium, calcium, boron, all of those macro micronutrients that a plant may need. And then as they have more bacteria, it becomes so concentrated in their bodies that they can do nothing but exude it or poop it out right at the root surface, providing that plant with exactly what they need at the right time. So I think that component of the dynamic temporal relationship of what's happening across all of that surface area, right? When you get deeper roots, every micrometer of that root surface has potential to host a community. And so then it just becomes exponential for what we're missing when it comes to plant health. Not to mention fungi, right? Fungi are the future. Fungi are everything. So carry on.
I'm cognizant of the need to move the conversation forward, but there's one story that I'd like to share that is so powerful.
Please.
A South African professor was giving a presentation on the control of Pierce's biocontrols, as in specific bacterial strains for the control of Pierce's disease on grapevines at a university in California that one of our growers attended. And so the grower was asking him at the conclusion of the presentation, he was describing the various effectiveness or lack thereof of different types of bacteria in controlling this disease. And our grower asked him the question, did you consider the possibility that perhaps there might be nutritional factors or a lack of nutritional integrity that might contribute to disease susceptibility? And he got the most amazing response. The professor said, oh yes, we have to run sap analysis on all the plants that are part of the trial. And we only use plants that are very low in potassium as a part of the study, because when plants have adequate levels of potassium in this particular environment, they're not susceptible to Pierce's disease, and we weren't successful in producing an inoculation. And it's like,
Yeah.
Here's
Positive forehead.
Great answer. That's hilarious. Yep. Oh, that's brilliant. Which actually is a perfect segue. So, attendees, I had also challenged all of our panelists to use this as a time to get to know and ask each other as experts what it is they would like to know. So we're going to start with Dr. Elaine's questions for John.
Well, how amazing that the first question is about sap analysis.
Hey, perfect segue. Thanks for setting me up.
So what testing protocol would you use, John, for sap analysis? How are you obtaining your sap, and what exactly you're going to send it into the chemistry lab to be tested for?
So the sap analysis protocol that we are using was first developed in the Netherlands by a team at a laboratory that is called Nova Crop Control. And today we are still sending our samples to that laboratory. There are other laboratories and other technologies seeking to be developed or in the process of being developed here in the States. But in our opinion, they still lack the data consistency that is needed for really optimal management. And so the protocol that the team developed in the Netherlands, which I completely back and support, is for longer duration, longer season crops like apples or tomatoes or soybeans. We collect a sample every 14 days through the growing season. From the moment the plant is large enough or has large enough leaves that we can collect a viable sample, all the way through until senescence, until leaf drop. And for rapidly growing plants, or for plants that are extremely high in value, a rapidly growing crop would be a spinach crop, for example, or a high value crop might be strawberries. We will double that intensity and collect a sample every 7 days. And for each of these samples, every time we collect a sample, we collect the last fully mature leaf on a plant, and we collect the oldest leaf on a plant that is still green and photosynthesizing. And we keep these two samples separate and send them to the laboratory separately. And then the laboratory measures these two samples separately, reports them on the same report. And this is incredibly powerful and valuable information, because plants will sabotage their foliage to support their fruit. And so when you see nutrient levels shifting and changing, perhaps the new growth at the top of the plant has 4,000 parts per million potassium, but the old growth, the oldest leaf that is still green, only has 2,000 parts per million. That is the plant's immediate signal to us that it is experiencing a potassium shortage, and it is sacrificing its older leaves to move potassium into the fruit, or perhaps into the new growth. And this is incredibly powerful, because all of a sudden it gives the plants the capacity to communicate to us exactly what their nutrition requirements are. And I've spoken at length in various webinars and presentations about the many things that we have learned from plant sap analysis, but I can just say that when you begin using sap analysis with a rigorous protocol consistently, it reduces fertilizer applications by anywhere from 40 to 80%, because growers see very clearly that they don't need it. The plant doesn't need it. Becomes clear very quickly that much of mainstream agriculture is consistently over applying most nutrients. And this is a very important point, because the majority of the disease problems and the insect problems that crops experience are not a result of deficiencies. They are a result of the excesses of the products the farmer applies. And so it's kind of a win-win. There's this, we sometimes get the reaction of it's too expensive, because pulling a set of 2 samples and sending them to the laboratory, shipping is expensive. We send them overnight air. And depending on how many samples you're shipping, it costs anywhere from $75 to $100 per set of 2 samples. And you do that every 2 weeks through the growing season, that amounts to a significant expense. And a significant expense that is a new expense for many growers. It's not a place where they're used to spending money. And so they look at this and say, well, this is too expensive. I can't afford this. But over and over again, we find that we reduce the inputs budget by 40 to 60% or more. All of a sudden, not doing the laboratory testing becomes really expensive.
Right. Yeah, I find that with farmers as well. They're very pragmatic and they're very economically minded. And if you're able to explain those things, then it helps that transition and thought process occur easier. Yeah.
Yep.
Brian, have you done some sap analysis as well?
I do. Yeah.
Cool.
I have a number of clients that we do SAP analysis with, and it is a very helpful tool. You know, just as John was mentioning, you get to see real time what's happening with the plant. And I definitely agree with you, John, about the excesses. You know,
My PhD research is jealous of your ability to do that. That would have been really fun. Wish I added that layer.
Right.
Sure.
So where did you
It is a
send
very
yours?
valuable tool.
Where did you send your samples for the SAP analysis?
You asking me or John?
I'm asking you, Brian.
Yeah, I have used a company. I have one of my clients have used John to do the plant sap analysis, and I have a couple others that have used another company in Oregon called Apical.
So John, do you do your analyses in-house?
No, we do not. We have
Okay.
you might refer to it as a virtual lab, if you will, called Crop Health Labs in Ohio. And what we do at that facility is we simply aggregate samples from all over the United States, and we send large packages of 400 samples at a time to the Netherlands by overnight air.
Yeah.
Right, you said that. Okay, that makes sense.
Well,
Cool.
I think the important thing too is that it's especially over time, as you're taking more and more samples from the same crops, you know, globally, I guess you could think of it in that sense, we get a better indicator as far as what really should be the appropriate amount of potassium, phosphorus, all the different, you know, macro and micronutrients in that plant. Because it, I mean, really farmers are used to that kind of paradigm, which is, okay, I should be here. I'm either too high or too low. How do I adjust it to make it to fit? And I think being able to have that kind of recommendations and that kind of ability to express your data in that way is very important.
Ryan, you raised another important point, which is it's common if it's common for us to just assume that if a technology gives us rapid results, then the best way to use it is to make rapid adjustments. But that's actually not the case. From my perspective, one of the most valuable uses of SAP analysis is for us to think and to time any necessary soil amendment applications differently.
Mhm.
So it's very common for us to observe that crops have a surplus, an excess of, let's say, potassium that is causing all kinds of nutritional imbalances and disease and susceptibility issues because the grower applied potassium perhaps 2 months too early. And then 2 months later, there is in fact a significant need for potassium if his soil doesn't yet have the microbial ecology, or perhaps it's a very sandy soil and there simply isn't an existing potassium base to pull from. There are cases where we need to apply some of these amendments, but the timing is absolutely critical. And so this is why it's so important to collect samples every 2 weeks throughout the entire growing season. Is you develop this curve, this graph of nutrient supply from the soil. And all of a sudden you realize that, wait a minute, I need to apply this or that nutrient soil amendment either significantly earlier or significantly later to time the nutritional release to match the crop's nutritional requirements.
Mhm.
Yeah.
What I would want to do is look at what biology we need to put back into the system so that you can step around having to put a chemical addition back into that system. How do we get the bacterial community or the fungal community or the predators of those microorganisms? What is the combination of what we need to do to achieve the same thing, but without use of the chemicals? Because the chemicals are always going to have a negative impact on
Mhm.
the microorganisms in the soil when we're putting them out as various kinds of salts.
Right, or balancing them so that the plant has what it needs to be able to produce the exudates so that community can establish and then eventually transition to what you're describing, Elaine.
Yep. And get the whole system working the way it's supposed to, that these organisms have been doing for the last, 3 billion years. Can we figure out how to manage that system?
Yeah, I think the aspirational ideal is to develop our understanding of microbiome management to the point where we can develop these microbial ecosystems that can supply all of our crops' nutrition requirements. And we also have to remember the context that there are some soil types across the country which don't contain the nutrients geologically. It's not in the geological substrate that the crop requires. And I'm thinking specifically of some of the very minute trace elements like selenium, for example, or like molybdenum that are required in only grams or ounces per acre concentrations. These are not universally present in some of our geological substrates. And these are also the nutrients, and this is something worth mentioning with the SAP analysis that we're conducting. We're measuring 22 different parameters, including cobalt and selenium and molybdenum and so forth. Actually, I don't think cobalt and selenium are commonly reported, although we do get to see some of that data. So I want to offer that as context that this is the ideal that we need to strive for, but there are some soil types in which we need to address some of those cases where the trace minerals simply don't exist in the geological profile.
And they're few and far between, those kind of strange soils that don't have some of the mineral nutrients, especially where you have one tectonic plate diving underneath the other, and you collect something that's been at the bottom of the ocean for the last 500 million years. Those are odd soils. And so when we know that we're dealing with that, then we've got to come up with a way to gently bring back those missing nutrients. Yeah, so haven't had to deal with those that much. So
I
yeah,
think there's
some things I don't know.
Yeah, another element to add, Elaine, and this, I'm only speaking from the experience, the data that we've collected. I'm not a geologist, so there's a great deal that I don't know. I think my perception is another factor that has occurred in recent history is that our topsoils have become severely degraded or eroded to the point, or weathered might be a better term, to the point where some of these elements are no longer available to crops in the A horizon. And so when we look at the map of selenium adequacy, for example, there are parts of the country with lower rainfall where we have high selenium levels, and other parts of the country that are chronically selenium deficient. And it's probably, I would be comfortable with the expectation that there's a reasonably good probability that the selenium does exist in the geological B and C horizons, but it needs to be brought up and be made crop accessible. And that is obviously a process that takes time. So
What you need is to get the roots of your plants down into those profiles,
Right.
along with the biology,
Exactly.
that will make the enzymes to pull those nutrients and turn them into a plant available form. We have selenium in all of our agricultural soils. The selenium might not be present as soluble selenium, which is what the plant needs, but we've got adequate levels of selenium in that profile. What we're missing is the
Yeah.
biology to do that change of those nutrients from not available to plant available.
Well, I agree with you, and I think it's a combination of two things. What we're missing is we're missing the biology, and we're missing the plant capability. And part of the reason we're missing plant capability is because we're missing biology. But there's another factor for plants as well, which is um, if they don't access the selenium or molybdenum or whatever we're discussing, it makes it that much more difficult for them to access it in the medium to short term. And so what I've observed in our experience is that if we then spoon feed just enough selenium or molybdenum or cobalt or what have you, we can stimulate plant health to a much higher degree and produce much more abundant biology. And it just kind of, it speeds up the overall regeneration of the system. It makes it a lot faster.
Well,
And
and
one thing,
we can think about the actual chemistry of a crystalline lattice structure. The geology can be seen from a biogeochemical perspective or from a biology perspective, right? So this is the great paradigm shift of regenerative agriculture, which is not a new idea by any means to honor indigenous traditions around the world, but it is a paradigm shift that we need in our current culture, that is seeing the soil not as a geochemical substrate that the plant extracts from, but instead that the much, much older microbial community has a greater diversity and access to enzymes that have the capacity to extract out all those minerals, that then have relationships with those plants. So shift it, and one can see a soil that's limited in selenium from either perspective. From the biogeochemical perspective, it is that it is not there and we need to put it back. From the biological perspective, it is that maybe it is in lower amounts, but there's also someone somewhere with an enzyme that can make it more available to plants in higher amounts. But maybe the plant does need some help to get there. So in line with what you're saying, John, you're giving that plant the boost to be more robust and vivid and able to get those roots deeper, to reach more of that selenium. So then maybe there's a transition period. You end up changing those inputs over time.
in fact,
Yeah,
that's
it's just a
go ahead.
question of speeding up the process.
Mhm, mhm. That makes sense.
Yeah.
You know,
Right.
the way I describe this with my clients is that when we go into these endeavors, we're going to switch from conventional organic to a biological, type of paradigm. We're in a transitional period. We're in this transitioning from a massive dependency of the plant on the farmer to provide those nutrients to where we are allowing the microbiology to provide those nutrients and do the nutrient cycling is happening in the soil. And plant step analysis is a huge tool for me because the farmers aren't going to really allow for you to reduce production or want a reduction in production. And it's kind of like the whole analogy of trying to change the tires on a plane while it's flying in the air. So you've got to introduce the microbiology, but then also make sure that they still have the right nutrient balance to produce a crop. And you want to be able to minimize the amount of those excess nutrients that you're putting into that system. And I think Plant Sap allows us to be very specific and very targeted about the type of those additional nutrients that we put into the system. And so I find it an extraordinarily useful tool for the work that I do. That while I'm focused on trying to get the microbiology back in the soil and get an optimal soil food web, that I can work with the farmers to apply minimal amounts, specific amounts of certain type of nutrients to allow for that production in that crop to still occur. I hope that made sense.
Absolutely. Yeah, no, that's reminding me of something one of my CTP students, Simeon, has been actually working with Todd, who's here today, part of why I'm bringing it up. And the philosophy in that, in a mindset he's describing, is that you got to get the chemistry of the soil healthy, like friendly enough to support a microbial community. So if you just put a whole bunch of microbes into toxic selenium mines, you're not going to get everyone to grow, right? So this is something we consider as well. But there is a capacity for microbes to alter their chemical environment because every living being is going to make their own little home how they like it. So there's the biology. I also live in the camp where the biology drives the chemistry, but there is a certain amount of chemistry that's going to impact whether or not that microbe survives. So it's this balance of both, right?
So if I can reflect on this, I actually agree with what you are saying. And I think we're saying the same thing. So I want to try to clarify my perspective a little bit.
That'd be great.
I see this as an interaction between three different drivers of the system, if you will. Actually, one is not technically a driver, but there's three different pieces that need to be present for optimal healthy plants to be produced and to really drive regeneration of soil health very quickly. The foundational piece is really vigorous microbiology. On that, I think we're in complete agreement. The next question becomes, what is it that drives very large active microbial populations? And I would offer that what drives large microbial populations is an abundant food source, which is provided by really healthy plants, particularly plants that are photosynthesizing at higher levels or at elevated levels above what is considered to be common. And so this is a bit of a sidebar conversation, but I think an important one. Most of the plants and crops that we observe and interact with are photosynthesizing somewhere in the neighborhood of about 15 to 20% of their inherent genetic potential. And it's not reasonable to expect to get 100% because that would require a perfect environment with perfect lighting and carbon dioxide levels and temperatures, et cetera. But it is reasonable to expect to get to 50 or 60% or perhaps even higher in agricultural environments. So think about what that means. That means that if you move from 15% to 60%, you've just quadrupled the sugar production in every 24-hour photo period. But when you quadruple sugar production, it's improbable that you're going to quadruple plant biomass or going to quadruple yields. So the question is, where does all the extra sugar go? And we know the answer to that. It goes into the root exudates. And this is the fundamental difference. This is why there's this huge discrepancy in reported values of percentage of carbon partitioned out through the root exudates. The literature reports 7% to 80%. It's because of this variability in photosynthesis. So from my perspective, the driver, or maybe the better word would be the substrate or the requirement to produce these very vigorous microbial populations that we desire is really healthy plants. Mm-hmm. So now we get to the next question, which is, what is required to produce these really healthy plants that are photosynthesizing at high levels? And the answer is, they need two things. They need a healthy microbiome and they need balanced mineral nutrition. And this is, I think, the perspective that Brian and I have been mentioning is sometimes the way to hack the system or to accelerate the system is to provide the nutrients that the plant requires for optimal photosynthesis when the soil isn't yet supplying them. Even though the soil biology may be able to supply them in the future, it's not yet happening today. And so that's,
I agree.
Most of the time, we can meet that demand if we're putting in really good compost, which is the organic matter that allows this initial get up and go to start happening. It is the food that leads into getting the sugars into the plant, or getting the nutrients into the plant for the sugars, the whole metabolic processing happening.
And for anyone who hasn't listened to Elaine a whole lot, when she says compost, she's referring to not just the organic matter and things like that, but also as a vessel coated with microorganisms. So just so that we're all on the same page about that term, that's what Elaine means by compost.
Yeah, biologically complete compost that has the balances of the nutrients already present, and we make sure that we put the kind of compost into the system that's going to wake that plant up, get it going very rapidly, provide the nutrients. So yep, I think we've got that into the process pretty much figured out, and it doesn't have to be extremely complex. We just have to get out maximum diversity that we possibly can manage to get the plants up and started at the beginning of the growing season.
I think there's an important element here, which is with the technology we have available today with SAP analysis, we don't have to guess whether our soils and our soil and our plant microbiomes is delivering the nutrients the crop requires. We can measure it, we can validate it, and we can see.
And we like to do that analysis on the microorganisms. If we've got the bacteria and the fungi balanced, and we've got the protozoa and the nematodes and microarthropods in good numbers, I think we see that the plants are moving in the right direction. Now, I'd like to document that by doing a number of tests where we do take the plant sap analysis, and then we can fine tune what needs to be in the compost at the beginning of the growing season. So we set the stage to make everything work all the way through the growing season.
So what I'm hearing is that with our powers combined, we can take the massive database of plant sap analysis and start to understand the soil food web biology and match those two and just generate, we could create something incredible here.
I absolutely expect that would be the case.
Right. You know, I have a client where I'm kind of doing something similar in that sense, where we have decided to take a block where we're going fully biological. We're only using compost extracts, compost teas. And then other blocks, we're doing different nutrient additions as well as with the microbiology that we're trying to apply. And then looking at plant sap analysis to try to see some of those differences that are in there. And it's amazing to see that, you know, my farmer was pretty impressed that the biological only block still had a number of nutrient deficiencies and wasn't cycling nutrients as effective. But he thought when we went fully biological, it was like, okay, these plants are going to get really set back, and come to find out, well, it's maintaining nitrogen levels in the plant very well. You know, there's certain nutrients that it seemed to be, okay, yeah, this is cranking along. I'm not having to apply any of those, you know, fertilizers into that system.
That's a ton of fun.
It is.
So we have so many interesting questions coming in, so I'm going to push this conversation forward a little bit.
Sure.
But I do want to know, John, could you tell us just briefly, what is the difference between plant sap analysis and testing Brix? What do you learn that's different there?
They have no similarities to each other.
Great. Do you ever use Brix testing?
Not anymore. Not
Okay.
anymore. We used to a lot, but we don't anymore. And I actually wrote a blog post on the opportunities and the challenges with using Brix reading. So I would refer people to that. The foundational challenge is that while it can be a useful tool, it requires extensive experience and familiarity with it to be able to use it as a management tool.
Mm-hmm. Yeah, that's great. I'm going to leave it at that. If you want to know more, go read John's post. So I'm going to jump in because I am excited that we got a question from a couple of our students. And so here are some questions from Uta, who's one of our Soil Food Web CTP students. And she's asking, how can we support plants to secrete exudates that support mycelium growth in both annual and biennial crops, as well as perennials? So healthy plants, how do you three each think about that plant's life cycle?
I don't want to dominate the conversation with someone else. Like, go first. You want me to?
You can
chime
start
in?
us off if you're feeling it, John. You got the energy. That's good.
So my observation is that when plants reach level 3 of plant health, their fat content increases on from a dry matter basis to somewhere that depends on which plant species we're talking about, but fat content on a dry matter basis can increase from a baseline of 1.5% to 2% all the way up to being as high as 6 to 8%.
Mm-hmm.
And when that occurs, once plants reach this level of health where they have this elevated lipid content, there is a corresponding rapid proliferation of the soil fungal community, much more so than the soil bacterial community. And my understanding of that from Dr. William Jackson's book, Organic Soil Conditioning, is that this is because most bacterial species are not able to utilize fats effectively, and that fats are primarily consumed by fungi. And this is something certainly that Elaine, or probably all of you, would know more about than I do. But that's been our experience, is that you can actually help develop fungal populations on annual and biennial crops simply by elevating plant health to the third level of the plant health pyramid or beyond.
That's neat. Right. So when we talk about exudates, just to be clear, that doesn't just mean sugars. That's not just straight up glucose. It is largely carbon-based. However, that can also look like benzoxazinoids in wheat plants, which can then help that plant acquire iron, which it needs for its chlorophyll metallic cofactor. Again, photosynthesis is the most beautiful thing there is. So, yeah, lipid-based exudates are going to recruit and maintain a lot of those, because those lipids have a higher complexity and require different and more enzymes. Yeah, Elaine, does that bring anything up for you? The fungal to bacterial ratio of a soil?
What we see is that as we go through succession, we definitely see a greater and greater dependence upon fungi to grow the plants that are typically present in that stage of succession. Now, I've never tried to tie that to the amount of fat or lipid. So that's going to take me some thought to try to.
That'd be interesting, huh? Yeah, totally.
Track down the literature to support that.
And again, there's this interesting chicken and egg scenario, which you sometimes ask the question, which comes first, elevated fungal populations or in the soil, or elevated lipid concentrations in the plants? Because what we see happening is they're intertwined, because in order to get high lipid concentrations, plants need to absorb the majority of their nutrients in the form of microbial metabolites, which is where the fungal population and the mycorrhizal highway becomes so critically important.
Mm-hmm. Yeah, Brian, what have you noticed regarding mycelium growth, mycorrhizal specifically, or otherwise, just fungal growth in your systems?
I mean, for sure. When I start to see, the plant's production, the plant, and just that observation of the plant health is going up, you tend to see a higher microbial population in those plants. As we start to move that system along, it does kind of move along this exponential rate of growth. So it makes sense to me. I haven't, looked at as far as, just purely as far as the lipid aspect is concerned. But I definitely do see that correlation between plant health, microbial populations.
Yep, and it's like when we look at the benefit to the plant, if less than 12% of the root system is colonized by the mycorrhizal mycorrhizal fungi, and it is a plant that requires mycorrhizal fungi, that plant's going to be in a world of hurt. It can't defend itself, basically. As you get to 12%, you start to see some benefit from the mycorrhizal colonization. So you're getting more movement of nutrients that the plant needs moving into the root system, and it's affecting the plant's growth rate. Up to about 40% of the root system colonized, you continue to see improvements in the benefit to the plant. Above 40%, the only thing I've ever seen is the attitude is, well, it's kind of more is better, but it doesn't make that much astounding benefit to the plant. And so this conversation here is making me kind of rethink that, because what if after 40% colonization is where a real shift in what the mycorrhizal fungi are doing, and that shift is triggered by higher lipid levels or something. So lots to go back and think about.
Yeah. Yeah, it's definitely, this conversation has stimulated me to just,
It has stimulated so many interesting questions. So I'm
Yep.
just resetting our. Is this the right screen? I'm not sharing all of my
Yep, you're good.
background emails. Okay, great.
Yep,
Love
yep,
it.
you're good.
Mhm.
Yep.
Fun. I'm also not used to a sun, so I'm like in this jungle right here with this. It's super fun. So here's a question from Thomas. Can you talk about the efficiency of foliar applications to support plants until our microbial network and the soil can take over? And I think this is Thomas Clausen. I think I saw your question come in. Thanks for being here, Thomas.
I
So-
assume that, yeah, I assume this question is directed toward me only because I've been a very vocal champion of the use of foliar applications.
Yeah, tell us about it.
I think I spoke a bit to this earlier. I see foliar applications as, let me say it this way. When we have these large tractors that we use at pulling competitions, they have tremendous horsepower. They're really souped up. They are designed to be able to pull tremendous loads very rapidly. And this is an analogy for what our soil systems and our soil microbial systems are capable of. There is tremendous untapped horsepower that we can unleash. But in order to get those engines started, they require the use of either starting fluid. And foliar sprays are like that. They are an injection of a very high energy fuel that accelerates the overall system and gets the flywheel rolling a lot faster. So they are a tool to be used to increase specifically. I see the primary function, the primary purpose of using foliar applications is to elevate photosynthetic efficiency from the baseline of 15 to 20% up to 45 or 50 or 60%. So that's the purpose of using a foliar spray, is to provide the plant whatever enzyme cofactors it might be missing to spike photosynthesis so that the plant can feed more soil biology.
So when you're spraying it on the plant, just walk me through, how is the plant actually, is it going in through the stomata? What's the imagination there?
No, it can a small amount, but the majority of plant absorption, I actually have a couple of blog posts where I talk about some of the various forms of absorption, but there is direct cellular endocytosis and absorption into cells. But there's actually another mechanism which is just beginning to be recognized and studied in the literature, which is that we have this microbial population on the phytoplane, which bears many similarities to the microbial ecology in the rhizosphere. And where we can put on these nutrients and we have various bacteria and yeasts on the leaf surface that actually metabolize them and absorb them. And they have a symbiotic relationship with the plant, same as the bacteria and the yeast and fungi in the rhizosphere. And so they provide nutrition to the plant in exchange for various sugars and exudates on the leaf surface as well.
I've been using this with a couple of my clients as well, where they're doing full applications, using plant sap analysis to get down to the, minimal amounts of these foliar applications to apply. But one of the things, we're always trying to look at is number of times we have to put tractors on the field and the labor involved. And so we combine our compost tea sprays with some of these foliar applications and have done some testing to see, does the products that we're putting in foliar, do they impact the microbiology when we do a compost tea spray? And for the most part, the quantities that we're talking about spraying foliar are small enough to where the impact the biology appears to be minimal. Although there's been a few products that we've tested that definitely, especially at the ratios, that definitely impacted the microbiology. And so I think that's more area of research to do as well, is that when we're doing the foliar applications, what is the impact potentially to the microbiome that's on the surface of the leaf, either being there already or being sprayed when we're doing compost tea applications, or applying them at the same time frame if we possibly can.
Brian, you raise an excellent point, and I actually taught an hour-long webinar on that topic that's on our YouTube channel. There are a number of characteristics, but you should, if you're combining nutrients with compost fuel, first of all, you should not apply a product that is damaging to the soil or to the phytosphere ecology anyway. And there are some nutrients that you have to be really careful with. Even if the plant requires them, you have to be really cautious with copper, for example, and manganese and boron, because those can all have biocidal effects in high concentrations. So for those nutrients, it becomes a question of concentration and also form. Are they chelated? And you can certainly buffer out some of the potentially harmful microbial interactions. So that's an excellent point. Completely agree.
Great. Fantastic. So let's move on to a question from Cerise. Do you believe that all crops have a place in sustainable agriculture, or are some crops simply unsuitable for a global sustainable farming system? What do you think, Elaine?
If some human being or some animal is going to eat it, I would suspect it's certainly have a place in sustainable agriculture. We can grow whatever we want to grow using sustainable or regenerative practices. So I don't think there's any crop that's really unsuitable if somebody wants to actually eat it.
Right, and I would also restate that as, like, is there a plant in the world that doesn't have relationships with the microbial community? And if that is the case, then maybe you're right. Maybe this kind of system doesn't work for them because they exist solely in that biogeochemical paradigm where you have to stick them in some kind of medium and then flush them with the right nutrients. But as long as that plant can establish relationships with the microbes who have all of that diverse suite of enzymes,
And
I think it's fair game for all plants.
I don't think there is a plant that doesn't or can't establish relationships with the biology in the soil, even weeds, you know, things that are growing where there's nothing but perhaps some bacteria or a few bacteria in that dirt because of some catastrophic occurrence, like, you know, a wildfire that the temperature is 600 degrees Fahrenheit at the surface of the soil and stays at that temperature for 48 to 72 hours. Try to grow a plant in that, you're missing the biology, and even weeds won't be able to grow. So when we start thinking of what came first, it's always the microorganisms in the soil have to be there to support the growth of that plant. And so we always have to have this biology.
I
Brilliant.
think
For me, I think it comes down to, the question is, maybe turn its head. It's not what crop you grow. It's more about how you grow a crop.
Have you
is
made
the impact.
sure that the balance of the bacteria, fungi, protozoa, nematodes are correct for that crop? You
Yeah.
want to grow a weed, you really want to grow that, then you better not have very many fungi in the system. Just is not going to happen. As soon as you want to get rid of the weeds, you just up the fungal biomass, because now you're going to have the proper ratios of nitrate and ammonium.
I think there are crops today that are grown unnecessarily and that are grown for the wrong reasons. Classical example being corn for ethanol production. I don't think that has any business being a part of our future.
I think we can agree on that one. Yeah.
I do think that our cropping systems do need to evolve to include diversity. I fully anticipate that crops of the future, with very few exceptions, will not be monocrops.
I sure hope that's the case. I definitely believe that multi-cropping is a huge thing.
So we got one clarification about this question, and that is, are there any crops that are too high maintenance to work with using the soil food web, or that would be too challenging in order to sustainably use these practices, or no?
Well, yeah, you're not going to grow tropical fruits in Canada without a greenhouse or something. So,
But you could grow those tropical fruits well using these practices in the tropics.
absolutely,
Right.
because the weather, the conditions, and the biology has already been designed to grow with those plants under tropical conditions.
I think there are plants and varieties today which have been genetically bred to no longer be adapted to a biological system. They have been weakened and they've been bred to be dependent on soluble applied nutrients.
Mhm.
So I don't think it's accurate to say that of an entire crop, but there are varieties, advanced modern varieties, which do not adapt well to the type of
Right.
approach that we're talking about.
Yeah.
Which is
Yeah.
why seed sovereignty is such an important component of maintaining that wild and genetically diverse capacity to maintain relationships with all below and above ground microorganisms, macroorganisms. Awesome. Great question, Cerise.
Yeah,
Thank you.
that was a good discussion. I appreciated
Mhm.
that.
All right, Robert M. I have some areas of my garden that are hydrophobic, probably from eucalyptus and acacia trees that have oils that cause a wax coating on soil particles. Anything you can think of that would introduce organisms that can help bring this down. I do try to be as fungal as possible in my entire garden. So how can we help Robert M. accelerate the breakdown of these waxy oils built into the acacia eucalyptus leaf breakdown?
Get some microorganisms that will decompose that wax coating. So, when you have the situation where you're watering your ground and the water just runs off in an area, maybe, 4 or 5 feet away, finally takes a dive into the soil. That area that is, there's a specific word for it and I can't remember it right now. But when it's resistant to the water being able to penetrate, that's where you've got to get these fungi, late successional fungi that typically will deal with the hydrophobic compounds that are produced by eucalyptus, for example. Acacia trees, not so much, but there's a banana leaves are one of my favorites because boy, I had fun trying to figure out how to compost banana leaves because they're coated by a layer of wax. But as soon as the fungi get through that layer of wax, now you're into the green part of that banana leaf. And in fact, it's concentrated so much nitrogen that it's a high nitrogen source. So you have to look out for that. You have to, do some practicing on all of the plant materials to figure out, do you have the fungi that will decompose the waxy leaf material?
Right, it's tricky when a starting material going into compost can be both complex and also punchy party food that can fuel a lot of growth that's very rich in protein or lipid, like fish hydrolysate, right? Is complex but good fuel. So,
yep. And it's kind of a surprise where after you've had your pile just sitting there going, no, not going to increase in temperature. We're just sitting here doing our thing. And you keep putting on more high nitrogen and more high nitrogen, trying to get it to go. And all of a sudden, the fungi finish doing their wax job and boom, you have instantaneous combustion. So look out for that.
So Brian, I know you spent some time in
one thing
California. Did you have any eucalyptus leaf trouble? Or
No,
whatever it is you were going to share?
No, well, I was just going to actually kind of, yes, we're on the same thought process here. I would, look at Robert's question and say the hydrophobic nature of the soil. I don't know. I mean, yeah, there may be some impact from eucalyptus acacia trees, but if you really have a good microbial community, well-structured soil and so forth, you shouldn't have any of those hydrophobic issues. So I would suspect it's not eucalyptus or acacia trees that are really causing that problem. It's probably something, more around, do you have a good microbial community set or do you have good structure built in that soil?
And if the fungi that you've got aren't doing the job on the soil and getting rid of that hydrophobic layer, you need to go searching for some different fungi. And we do say to people that, go find the closest natural area that has not been disturbed for a very long time. And you go in there and you take pinches, just pinches of some of the soil that's in there. Try to pick little bits of the really good fungi because you don't need a lot. Take it back home, inoculate it into your compost pile and get those really good organisms, get that increase in diversity going in your compost.
Or if it is the eucalyptus leaves themselves, then make a compost out of those eucalyptus leaves. And that those organisms that grow in the compost are going to
Yes.
be the ones that you're gonna are going to be your helpful buddies.
Got it.
All right, I'm gonna jump in. So we have a question. We have a couple questions from Curtis, but let's see. We have a few more in queue, so I'm gonna be a little brief here if possible. The broad question here is I want to transition into organic no-till row cropping. My question is, how should I start my transition? And this is probably something in general, transitioning to these regenerative processes. Where do you start? But the question specifically here, should I start with going for organic certification and use tillage to control weeds while I build up my soil? Seems counteractive to building soil structure, which I will just say, yes, you're right. There is a challenge in organic agriculture that requires tilling as an answer for a lot of those problems. But the tilling, no matter what, is going to slice and dice the fungi. So I'll just answer that one if you all have additional things you'd like to say. Should I take a slow transition to organic and use herbicides for the first few years while I build up my soil and delay organic certification? How would you recommend?
My opinion number two is you don't need to take a slow transition, and if you can remove the use of herbicides out of the equation, then do so as soon as you can. Especially if you're this is a transitioning, right? So they are transitioning to no-till row cropping. There are other tools you can use, woody mulches, perennial ground covers, other things you can do to help suppress weeds and those types of things. So those are the things I focus in on and get herbicides out of the equation as fast as you possibly can.
I like to say, leave all the asides aside.
Yes.
There are more than these two options. You don't only have to select these two options. There's not time to answer the question in full, so I'll just say get Rick Clark as a mentor, whether virtually or live. Look at what Rick Clark has done in transitioning 7,000 acres in West Central Indiana to organic no-till without the use of pesticides or herbicides through the transition process. Invaluable advice and real life experience.
Yeah. And I think people could come take the foundation classes that we offer at Soil Food Web, and you would combine the practices that would work for you most rapidly. I think it's very feasible to do the transition in one growing season. And if you don't achieve the end goal in one growing season, it's big. The people that we have those kinds of problems with, they won't listen. You got to make compost right. Compost is not just throwing a bunch of organic matter in the corner and waiting for it to decompose. You've got to make sure it stays aerobic. You've got to make certain that the organisms, the diversity of organisms are present. So a couple of things that you really need to think about when you're going to start making your compost piles.
Great answers. Love it. I'm excited. Okay, questions from Jack. Any recommendations on methods to terminate cover crops? Is the language
Don't
here.
grow cover crops in the first place.
Well, so hold on. Let me clarify. Elena's pro plants growing in the soil. She often will say not to use cover crops because that implies that then you're going to till it back into the soil or harvest. And the longer you can keep a healthy root in the soil, the more benefit. So
well, and
I just want to throw that.
Cover crops are always tall. They've tried to be selecting for the last, I don't know, ever since they've started doing cover cropping, basically. They're pushing for very tall plants because now you've got more biomass that you can till back into the soil, right? You can't grow a different crop because it's overshadowed by the cover crops that, those plant species that have been chosen to be cover crops. And that's, and I don't see the point in buying seed every year when you can choose perennial plants and you can go in and seed those perennial plants in, and for the rest of your life and your children's lives and your grandchildren's lives, you don't have to go out and buy seeds in order to maintain that protective layer that maintains nutrient cycling, that maintains the exudates for the bacteria, fungi, protozoa in the soils. So cover crops are just, come on guys, think this one through better.
Brian, how do you use cover plants, cover crops, etc.?
Like Elaine, I try to get my clients to move to cover plants and go into a perennial cover planting system. I do have a number of clients though that are, you know, really down the cover crop route, and that's what they want. Typically for me, it's crimp and roll is probably one of the best ways. That way you can create a layer of mulch, and you apply compost extracts on top of that. You get it to break down and go into soil profile. Other ways to do is grazing as well. That's another good way to be able to really cover plant, or sorry, cover. But like Elaine said, my philosophy matches very similar to hers, which is I try to get my clients into a perennial cover plant kind of system.
John, do you work with folks in managing how they protect their soil or grow plants?
Yes, in all types of ecosystems and all types of contexts. So we use annuals and perennials, and we manage them in lots of different ways. But in a broadacre commodity crop context, then I would advise the use of cover crops and crimping. And again, I would refer people to Rick Clark and Dave Brandt and other very successful examples.
Awesome. Yeah, and I've heard Dr. Tim LaSalle, who co-founded the Center for Regenerative Agriculture down over at Chico State. He also just speaks beautifully about the multi-species cover crops, in that case, cover plants or cover crops, but also having multi-species in there as a component to the whatever living root is in that soil, can also contribute wildly to maintaining a diverse microbial community. So I'm gonna pop out of our questions, just so that we get a minute in the last 15 minutes for us to circle back and make sure that this conversation completes itself in all the ways it needs to. So Brian, what questions do you have for John and Elaine?
Oh, I did have I have a question for Elaine. Let me just pull this up here real quick. I want to make sure I got it accurately here. So Elaine, what are the pros and cons of the emerging biological amendment market? Is there a standards regimen that needs to be created / enforced? Because that's one thing I'm starting to see an explosion of, is just all these different biological products that are being, you know, marketed and sold. And it feels kind of like the old Wild West snake oil salesman kind of paradigm. And it's very difficult from my standpoint being a consultant, because I get bombarded with, what about this product? What about this product? What about this product? It's like, oh, okay. Each one of those takes a little bit of time to research to be able to go through that.
Yeah, and so, you know, typically if there's a limited number of species in their container of bugs, I will indicate that it's probably not worth it, because a one-time application should get those organisms back into the system, and most likely they're actually already in your soil, unless you've been just crazy with the pesticides and inorganic fertilizers and etc., killing that biology in the soil. Additionally, you have to look at where those microorganisms were actually isolated from. If you're in Oregon and you buy something that the organisms were isolated in Pennsylvania, for example, those microorganisms are not going to be competitive in an Oregon community, in the soil here in Oregon. If you've got something that was isolated in California and you put it into the ground in Florida, you're wasting your money, because those California organisms are not capable of dealing with the climate, rainfall events, the temperatures, the seasonal cycle. Waste of time, because they're not going to be there. Probably within 2 to 6 months, they're goners. And so you want to be getting the local indigenous sets of microorganisms, and you want to get not just bacteria. How many species of bacteria should be in a good, healthy soil? And, we're probably up in the order of, it could be as high as 500,000. And so you want all those organisms in your soil so that all through the whole growing season, with the different ratios of temperature and moisture and sunshine and cool and hot and all of that, that you have somebody that will wake up and do the job that your plants require under the conditions that change all the time as you go through a growing season. So those are the organisms that we want. How many species of fungi are there in a good, healthy soil? We're probably looking at 100,000 or more. We actually don't know those numbers precisely because we haven't identified all of the different organisms, the bacteria and the fungi in the first place. So it's every year somebody else comes up with a higher number, and the year later it's another higher number of the number of species of bacteria and fungi. So
In their magical solution, right? Yeah.
or even in most of the time in their what they're trying to put on, what they're trying to sell you is only 2 or 3 species, and you're going to put that into something that already has 100,000 species. That's ridiculous. You're not going to get benefit for very long at all. So buying bugs in a jug, no. Go out and find them in your own bioregion. And that's what you want to buy. That's what you want to get hold of. Get that
Yeah.
into your compost, let it grow.
John, what's the snake oil version in your
I can
world?
apply the snake oil version.
Uh-huh.
Well, I have a different perspective, I suppose, and we have we don't manufacture any inoculants, although there are commercial inoculants that we do use on many of the farms that we do consulting work on. And we have been able to observe pronounced improvements in plant nutrient absorption from a single application upwards, and not just nutrient absorption, but also changes in soil chemistry for as measured by a soil analysis for upwards of 10 years after a single application. So I think they can be very effective. And these were inoculants that just contained a very few species. And so I do understand your perspective, Elaine. I've been trying to, for a long time, I had the question, why is it that some of these inoculants are able to produce these responses so consistently across such diverse geological terrains? And I think my hypothesis of one possible answer is that perhaps we should not be thinking about some of these available inoculants as a source of specific strain of bacteria, but instead of a source of genetic information. So we know that when we have a bacteria that develops antibiotic resistance genetic information, for example, we have this very rapid horizontal gene transfer where that information can spread throughout the entire soil microbial population very, very rapidly. And perhaps it's that type of genetic information sharing that allows some of these inoculants to be so effective. With all that being said, I am an advocate for the use of compost teas and more diverse microbial solutions. And in my professional work, unfortunately, I end up working with many large-scale farms who don't have the resources, both in terms of labor and raw materials, to produce compost and compost tea, and they are looking for something that they can apply easily in the form of bugs in a jug.
I think this is a challenge that we have in the community at large, which is there are very few producers of locally made biologically complete compost. And I think as we are able to get, a lot of compost producers having a focus on creating biologically complete compost, then we will have a very ready source that can be used in a lot of the agricultural systems. And right now, for some of the farmers, I can imagine, it's like all the compost I have is 80% manure and 10% or 20% bedding, and the diversity of species is really low and the compost is pretty crappy. And so then they would go to a bugs in a jug type of scenario. And so I think as we start to see a much more proliferation of good compost being created, that will be a major game changer in that community.
Yeah,
And we're trying to fill that gap, train the people that will be able to make that compost. And then the local people realize that you've got a great product and they don't care how much it costs because it's more than worth it, because they get all of these benefits when they get this biologically complete compost into their gardens, into their agricultural areas. So we can be selling it for $1,000 for a cubic yard.
Yeah, and
So I
one
know
of the big
getting to the-
barriers for widespread adoption of these methods is just the notion that to any farmer, the term regenerative agriculture, or to any of us here, we may have a different notion for what it means to do regenerative agriculture. So I think it's one of our big tasks and responsibilities as some of the leading voices in this conversation right now to make sure that we're clarifying the most important parts about, yes, we need the diversity and also the roots in the ground, and also and actually we have a blog post coming out for the Soil Food Web school that's all about the barriers that are keeping farmers from really being able to adopt those methods. Thanks to our brilliant communications staff, we have a lot of great people supporting us here. But we have a few moments left, so I want to turn to John's question, which I think is really interesting. He gave me a little preview of it earlier.
Yeah.
So
Don't want to lose sight of it. So Brian, I guess I'll direct it to you first. What is an opinion that you have or perspective that you have gained that is contrary to most of your colleagues?
I wouldn't say this is contrary, but is maybe not prioritized, essentially, which is, I think there is a very good collaboration needed between the high-tech industry and the agriculture industry, especially around the biological aspect of it. In particular, I'm looking at analytics. There's so much data that we have out there that really strong data analytics could help provide insight to these very complex systems. And I know people are looking at that, and I know it's not something that says it's not being used, but I think there's more of an interaction that could occur between that community of data analytics as well as what we're doing in the biological side of the house, and probably some very good learnings or understandings could be achieved from that.
Thanks, Brian. Elaine,
Yep.
what would be your response to that question?
I'd have to ask you what your definition of colleagues and
Well, some people have no peers, as we know, but what's your heretical point of view?
I have introduced quite a few heretical concepts in the world of soil biology and how important it is, because I like to bring up the story of when I started my PhD work at Colorado State University, and my major professor said, go talk to everybody in the university that has anything to do with growing plants and ask them whether you should be looking at your PhD project that we had thought up, which was to try to understand the role and function of fungi in the soil. And I went around to all of these people and to a man, and I do have to point out, it was only men I was speaking to in any of these professorships, but every single one of them all said, no, you absolutely do not want to do that research because you will not be able to find a job when you get out of school. When you finish your PhD, that's it, it's a dead end. And so we strongly urge you to not take on that research project. And it was a little frightening as a graduate student, thinking about, but this is what I want to do. I want to understand why fungi are important. And all these people had said, fungi don't do anything. Bacteria, they're not important. It doesn't matter that they're there or they're not. So I went, yes, from the very beginning, I went upstream instead of downstream. So yeah,
That's
I
brilliant.
have
Mhm.
been
Thank you
through
for
it
your
all.
heresy, Elaine.
Well, I was going to say, John's question relates directly to you, Elaine, because I think you basically have kind of pushed this, biological farming, everything to its paradigm, right? That I think you've really been the person that's been outside the box thinking and really kind of pushing it into the right spaces.
Mm-hmm. And if I could answer my particular heresy, it's interesting because my colleagues have kind of shifted over time as I went from being an academic to working amongst very brilliant humans who don't spend their time thinking about p-values, which is the statistical like magic thing for if that doesn't mean anything to you. But the particular heresy that I think I am trying to bring is that if we want to work with fungi, which is that incredible network that allows trees to support one another and connects the entire microbial community and provides structure, which is enough airspace for the water to flow deep into the soil. If we want to work with fungi, we need to behave like fungi. And that means connections. That means resource sharing. That means diversity. That means we need to incorporate ancient living communities that have worked and lived in symbiosis with nature for longer than my ancestors have been in the United States. And I think that focusing on and making sure that the voices that we're listening to includes a greater diversity is one of the things that I will continue to push for.
In the spirit of that, I think I echo so strongly your sentiments for the need for connection and the need for all of us. We need to develop our own peer networks to support us and to encourage us. And then we need to also provide that support and encouragement to a community around us. And that is really what is going to be needed to create the collective change we want to see in the world. And in that context, I would invite all of you to join a platform that I developed called Kind Harvest, which is perhaps the simplest way to describe it is Facebook for agriculture, but without all of the crazy overlord stuff. And where the desire is to facilitate good conversations, and the community that we have an awesome community there. And that community needs to hear from all of you in your work on compost and the soil food web.
Absolutely.
Yeah.
Have to go check it out.
Yeah,
That's
exactly.
brilliant. So as a
Yeah.
final punchline, John, what do you believe that is possibly heretical or counter to those you find yourself surrounded by?
Well, there's a few. One is I believe most of our problems are self-created, which is not a popular point of view. I also believe that the fastest way to change the system is to optimize plant nutrition with foliar sprays and microbial inoculants. And I know that that puts me in a different group a little bit from the work that you are doing here. But that's the heretical belief that I have.
That's brilliant.
Well, we're putting in all kinds of microorganisms every time we apply any form of compost. So
yeah, and we're trying to
microbial-
get the foods right for the organisms, right? So that's part of the, what do we feed our microbes? What are we feeding our plants? And who is there to participate? So it's
I don't
all
think
connected. Yeah.
we aren't that far apart. We're actually probably fairly overlapping in a lot of the things that we do, and we should work together.
Sure.
Can't wait. And thank you to all of our participants. Thank you to all of our panelists. Each of you, I think, brings a really, really valuable perspective and wisdom. And thank you to the folks behind the scenes. Sammy, Alex, Heather, y'all rock. Thank you. Thank you, participants. And yeah, if your question wasn't answered, we'll do our best to follow up with you. And thank you so much for contributing your thoughts and being a part of this community.